Well, good afternoon and good morning maybe for some of you, and welcome to the third in the series of New Cells Biotech webinars, highlighting and discussing the latest developments in 3D cell-based assay technology. I'm Dr. Mike Nichols, the CEO of New Cells, and I will be chairing today's webinar, which will discuss the application of induced pluripotent stem cell-derived models of the upper airway for the in vitro study of SARS-CoV-2 infection. Before I introduce our speaker, I would just like to remind you that we will be taking questions at the end of the session, and you can use the chat facility on the webinar platform to pose your questions, and I'll be collating them, and Lyle will be answering those, as I said, at the end. So, it's my pleasure now to introduce today's presenter and my colleague, Professor Lyle Armstrong. Lyle is Professor of Cellular Reprogramming at Newcastle University here in the UK, and also CSO of New Cells Biotechnology, which he co-founded with me over 5 years ago now. Lyle has researched in the field of iPSCs for over 14 years and has published widely on the application of iPSCs in the development of models of a variety of human cells and tissues for applications in drug development. So I'll hand over now to Lyle, who will take us through the presentation.
Wonderful. Thank you, Mike, for that introduction. So today, what I want to discuss is the work that we've been doing on making human upper airway models from induced pluripotent stem cells. Now, the title slide indicates that we will be talking about SARS-CoV-2 and the current pandemic, which is absolutely true, we will be. But before that, I want to discuss the rationale behind why we became involved in generating a model of the upper airway in the first place. Now, just a little bit of background about New Cells Biotech before we do that. Essentially, as Mike says, we were founded 5 years ago with the aim of producing 3D models of human tissues from induced pluripotent stem cells. Now, our particular focus is on modeling epithelial tissues. And the reason behind that is that there was significant or key expertise within my academic group, which fed into the founding of the company. And of course, we had substantial grant funding to allow us to develop those models in the first place. But our principal reason for investigating these tissues is that quite often epithelial structures are the first point of contact or entry for drugs and xenobiotics into the human body. So if there are any toxic responses or any physiological responses going to occur, it's likely that they will happen long before other organs. So the areas that we're particularly interested in New Cells, of course, are the human lung, kidney, liver, and of course the sensory epithelia being the retina and the inner ear. Primarily because there is enormous demand for those. There are a significant number of extremely burdensome diseases which affect the lungs, namely things such as asthma, chronic obstructive pulmonary disease, bronchitis, viral infections, pulmonary fibrosis, to name but a few. And I hardly need to remind anyone of the numbers of sufferers that are subject to these conditions globally. Similarly, in the liver, we have hepatitis, fibrosis, various infections of the liver, which create a significant burden on our healthcare resources. Acute kidney disease kills many people globally every year. And of particular interest to us, again, we're looking at macular degeneration, which this year alone is predicted to reach about 196 million sufferers. So this means that there is a substantial demand for new therapies for these diseases, which in turn filters down to us and translates into a need for better and more accurate in vitro disease models. So, getting back to the lung. Initially, we were approached to work on this by a consortium called IN3, which is an EU-funded group of industry and academia-based research groups who are interested in developing iPSC-based models for human disease, but primarily for advancing those as a toxicological tool. So we were charged with three things. Could we look at the damage of lung epithelia by xenobiotic substances in a 3D in vitro model? Could we advance that model to understand the transcriptomic and metabolic changes induced by xenobiotic substances? And could we then describe the consequences of those both changes, such as fibrosis, metaplasia, or the inflammatory components that lead to damage to other tissues, such as the vascular endothelium? Well, why did In3 particularly want to do that? The section which they asked us to look at was primarily the impact of nanoparticles, such as you would get from diesel and gasoline exhaust, on the lung epithelium. Now, there are a few things that those particles can do. Obviously, they're quite small, so they can integrate into the cells, into the surface epithelial cells, and that has several effects. Obviously, the cells can die, but assuming they don't, then it will bring about certain changes which are still quite harmful to the lung. The epithelial cells can secrete damage signals, essentially cytokines and chemokines, which can induce fibroblasts in the interstitial space to become myofibroblasts, which is the first step on the route to pulmonary fibrosis. Or you can recruit innate immune cells, such as activated or alternatively activated or classically activated macrophages, which in turn secrete their own profile of cytokines and chemokines, which cause inflammation of the lung and further contribution to fibrosis. So, what we wanted in our model was to be able to model the ingress of particulates of this particular type, but also to understand what the transcriptomic consequences of those ingresses were. So, for example, if we look at what, say, carbon black or carbon nanotubes, or asbestos do over a fixed time frame to the lung in vivo, we can see that there are certain transcriptomic changes, certain changes in gene transcriptomic changes. So we must be able to recapitulate at least some of that change. And if we look at cigarette smoke, for example, in other instances, you're achieving much the same sort of aims. It causes damage to the epithelial cells, secretion of cytokines and chemokines, ingress of immune cells, ultimately fibrosis of the small airways, or destruction of the alveolar walls, a condition that we would call emphysema. Of course, one of the things that we would also like to be able to do with the synthetic lung epithelia that we make is to understand how various drugs or various xenobiotic substances can induce metaplasia in the basal cells and lead potentially to lung cancer. Also, we're very interested in drug toxicity. In fact, this was our primary aim for building a lung model in the first place. For example, we know that drugs such as methotrexate and tyrosine kinase inhibitors are actually quite good at producing pneumonia, drug-induced pneumonia. Basically, the inflammation of the lung parenchyma and release of fluid into the interstitial spaces between the alveoli, which interferes with the gas exchange capability of the lung. In a similar way, pulmonary edema, which is accumulation of fluid in the air sacs, does essentially the same thing. But also a lot of drugs such as amiodarone, busulfan, bleomycin, etc., will cause the particular type of damage to the alveolar epithelium, which can lead to the onset of fibrosis. So all of these things we must be able to incorporate into our model. Now, of course, the gold standard in previous times was to use an animal model. And there are many advantages in doing that, assuming that we can find robust and reproducible ways to get the xenobiotic substances into the animal's lung. One of the great advantages, of course, is that since it is an in vivo system, we can understand at a gross level what the xenobiotic is capable of doing to, not just to the lungs, but to other organs within that animal. And you can look at the gross histology of those sections of those tissues to see which particular cell types are affected. You can measure reactive oxygen species, you can measure epithelial cell damage, damage signals such as cytokines again, ingress of innate immune cells. But the great problem with doing that is that the response of an animal lung to a xenobiotic substance or to a viral infection is often very different to that which we would expect to see in a human. So data that we would get from animal models has to be checked with some degree of caution. And of course, quite apart from the ethical perspective of using a living creature to test out something that may not be of direct relevance to humans, such as an industrial chemical. So, what about in vitro models? Currently, there are a number of approaches that we can use to do this. There are several commercial aspects. One of these involves the extraction of primary human cells from the nasal passages, the trachea or the bronchi. Those can be grown in vitro culture and exposed to an air-liquid interface to allow them to form a fully differentiated airway epithelium. Now, one of those is a product called Mesoscale, which comes from a company called Epithelix. There is a very similar model produced by MatTech called airway. But the disadvantages of using primary tissue are that it's twofold. We can't make so many constructs from the same individual because obviously being somatic tissue, it is subject to a proliferation-related decline in capacity. So your ability to produce mucilaire constructs from, say, one individual is limited. Now, that means, of course, that you have to use multiple donors, and that leads to the possibility of variation in the product in terms both of the genetic variability, the background genome of the individual donor, but also what their lungs have been or their airways have been exposed to throughout life. And I don't just mean were they smokers or, did they take a particular type of pharmaceutical product. The level of pollution that has been that is ingressed into their lungs over time can make a difference to the functionality of the cells that would be used to produce the construct. So, okay, one of the things that we do, the primary thing that we do in new cells biotech, of course, would be to make induced pluripotent stem cell lines from an individual donor. And that's great because we can, at least in theory, make constructs or products which are less variable. We can achieve, a much greater degree of batch-to-batch similarity between lung organoids or lung constructs that we can make. But until the last few years, the possibility of making something that was a broad equivalent of, let's say, airway was more restricted. Sure, there were ways of making 2D differentiation cultures, publications that describe that, or to make iPSC-derived spheroids. And those were interesting in that they did express some of the markers suggesting that we had, say, goblet cells or alveolar type 1 cells present. Or in the 2D differentiations, we had club cell markers. But the problem is those structures are not arranged into an airway epithelium in the way that these primary derived products are. So our aim was to change that. So the lung is, in some respects, quite a complex structure because the types of airway epithelia that we need to model changes as you go from the proximal to the distal airway. So in the proximal airway, basically the trachea and the bronchi and some of the larger bronchioles, you have a much greater predominance of these things, of airway basal cells. Then you have a number of ciliated cells, club cells, and mucus-producing goblet cells. As you go down the airway, as the airway branches and becomes more complex, at the border between the proximal and distal airways, you begin to have an epithelium of slightly different structure. It has more club cells, for example, and fewer basal cells. And it doesn't have any neuroendocrine cells, such as you would have in the upper airways. As you go down into the alveoli, of course, the situation changes completely. You've got alveolar type 1 cells, which are the epithelia involved in gas exchange, and what is the progenitor cell population of the alveolus, basically the alveolar type 2 Added to that, you've got various stromal components such as fibroblasts, smooth muscle, myofibroblasts, vasculature, and innervation. So our model was aiming to produce initially a construct which was architecturally similar to the upper airway. So we did this by establishing what is now one of our IP protected protocols to make bronchial epithelial basal cells from induced pluripotent. We can grow those to very large numbers, we can freeze them, we can bank them, and when they're thawed, they work very well at an air-liquid interface to produce a pseudo-stratified epithelium. We're quite lucky with lung in that it basically assembles itself. Some of our other models, we have to either, put various layers of cells down or even possibly 3D print them. But with lung, we're lucky. It does the job itself. What we don't have in the current model is representations of the vasculature, so no endothelium, no stromal cells, and at the current incarnation of the model, no innate immune cells. But we can use that as readouts to look at function and morphology of cilia. We can look at transcriptomic changes in the whole organoid, cytokine production, markers of oxidative stress when exposed to xenobiotics, DNA damage, and cell viability. The way in which we make the basal cells is an adaptation of literature protocols using a specific regime of growth factors, which basically sends the iPSCs through definitive endoderm via anterior foregut endoderm into a basal-like cell population, which we would normally begin to isolate at about day 14 and then grow up on a feeder cell population with specific media additions to expand very large numbers of the basal cells. Essentially, we can produce as many of those as we want and bank them. We can characterize them just on the basis of markers such as integrin alpha-6, NGFR, and cytokeratin 14. If we see those markers, we're fairly sure that we have a basal cell population. We can analyze them by immunohistochemistry, and we can compare them to comparable cells that would come from the primary lung. So, for example, we can see that we've got NGFR expression, delta MP63, cytokeratin 14, and integrin alpha-6. And there is a fairly good comparison between cells that we can get ex vivo. Air-liquid interface differentiation is relatively simple. We would grow the basal cells as a monolayer on the 0.4 micron PET membrane in a transwell insert. For about 3 days, it will take to become confluent. Then we change the medium, raise that to the air-liquid interface, and then the rest just does it itself. It will assemble intermediary cells, mucus-producing cells, and functional cilia, and above all, mucus. We can see the mucus by about day 60. We begin to get these swirls of material. And the fact that it arranges into these swirls implies to us that there is at least some degree of active ciliation in these cells. We know that they're forming a relatively tight junction monolayer because we can see increase in transepithelial electrical resistance over about the 60-day differentiation protocol. And if we wash those structures with PDS, we can remove the mucus buildup and then fix and section those cells so that we can see that they do have cilia on their surface. We can analyze the structures further. We can look at synaptophysin, suggesting the presence of neuroendocrine cells, which again characterizes this more as an upper airway model. We also have club cells, goblet cells, and basal cells arranged in the correct architectural location that we would expect them to occupy. It's important to know that the cilia are indeed functional. We can see the formation of tight junctions on the basis of Z01 staining. We can detect cilia on the basis of acetylated tubulin. But we need to make videos of those, and for this we're very grateful to Amsterdam University, in particular Jelle van der Boer, who helped us to make these videos of beating cilia. Now, one thing is true of the iPS-derived model, that the areas of ciliation in the cells are less than we would expect to see in models derived from primary airway basal cells. But it's very encouraging that the beat frequency of the iPS-derived model is broadly similar to the ones that we would make from primary cells and also from in vivo lung, with a beat frequency of about 15 Hertz. So we think that the cilia are quite functional. If we look at a video of those, we can see the cilia beating back and forth, and also we can do that at half speed to get a greater indication of how many cilia are present. So we have a model which works for ciliation, for structure. What we need to know is how similar that is in terms of its gene expression profile, particularly for cells which are, or for genes which are involved in xenobiotic metabolism. Now, if we compare an RNA-seq experiment, which we performed on airway constructs made from induced pluripotent stem cell-derived basals and primary-derived basal cells, we can see that there's a not unreasonable correlation between the transcriptomes of those two types of construct. And if we look at that in a little more detail, this heat map shows the levels of or relative levels of gene expression of three replicates of our iPS-derived constructs compared to three replicates of primary-derived. Now, of course, we can see that there are some differences in the expression levels of these individual genes. There could be several explanations for that. This could perhaps just simply be the genetic background of the individual donors of the primary basal cells and the cells that we use to make the induced pluripotent stem cell line. But if we look at that in slightly more detail, we can see that the enzymes that we were particularly interested in, such as phase 1 CYPs, phase 2 transforming enzymes, we can see that with a few exceptions, there is a reasonable level of correlation between the expression of these enzymes. And the same occurs for ATP binding cassette family genes and other solute transporters. So, from those data, we feel relatively confident that the iPSC-derived constructs are going to be useful tools to understand xenobiotic metabolism in our lung model. Now, I did promise about COVID-19. By the time the pandemic was declared in March, we were already quite interested in the idea of rolling out this product in the near future as a means of understanding drug interactions with the lung. But, never let a good crisis go to waste. Along came SARS, and we thought, well, okay, let's have a look at how our lung responds to infection by that particular virus. There is no need to explain the rationale behind that, given the continually increasing caseload of this particular disease. But there are two things that we need to know. One, can we replicate the mechanism of SARS-CoV-2 infection in our model? And for that to take place, there are a few things that we need to know. SARS comes along, a particular glycoprotein called the spike protein sitting on its surface, binds to the angiotensin II converting enzyme receptor, but it also requires the presence of a protease, TMPRSS2, to cleave or activate that particular glycoprotein on the surface of the virus. Once that's taken place, it's able to internalize and will become encapsulated in endosome-type membranes. Once that event has taken place, the viral RNA can be uncoated, can begin to replicate, but also can be transcribed to produce the polypeptides, which are going to be responsible for production of more spike glycoprotein, membrane proteins, envelope proteins, and all the things that the ER and the Golgi need to be present to assemble new virions, which can then be exocytosed from the cell. Now, of course, that has certain unfortunate consequences for these cells, which are just generally epithelial cells. They could be in the lung, or they could be in the kidney, or the liver, or many other tissues within the body. But let's stick to the lung. It's going to come along, it's going to infect the epithelial cells, and they are going to express their dissatisfaction with this process in much the same way that they would with any other damaging event. They will start to secrete cytokines, and those cytokines in turn will recruit immune cells who will begin to secrete their own panoply of cytokines. And this is a process that we have become to know as the cytokine storm, which is one of the significant problems in COVID-19. It leads to fluid in the interstitium, and long-term may lead to the induction of more fibrotic type events. So the big question, of course, is can our iPSC-derived model recapitulate these processes? The first thing we need to be able to show is the presence of ACE2 receptor and of the TMPRSS2 enzyme. We know that they're there. We can see them by immunohistochemistry. In fact, if we look at the expression or the staining of that particular protein on several of our other models, such as our hepatocyte models, podocytes, proximal tubule, we can see that they're all there. So they all have the potential to be infected by SARS-CoV-2. To some extent, our iPSC airway model expresses more ACE2 than our primary airway model, but that again could just be simply due to the genetic background of the individual that we use to make the iPSC line. So it's got the capability to be infected. Does it actually infect? Well, it does. Now, I have to stress that we did not do this work within New Cells Biotech ourselves, because we don't have appropriate class 3 containment facilities, and the last thing we want is SARS-CoV-2 lurking around our laboratories. So we set up an agreement with the Liverpool School of Tropical Medicine, and they were quite happy to perform infection of constructs that we sent them. Now, if we measure, say, a couple of hours after the infection event, we can see that there is some degree of virus present, but that could just be the initial inoculum. But if we look 48 hours later, we can see that the total level of viral particles based on platforming units in the cells is much greater than at the 2-hour period. So that implies that the cells are capable of receiving the SARS-CoV-2 virus and replicating virions. However, we needed to know if the virus had actually left a footprint present in the cells of our construct. So we did staining for the SARS-CoV-2 spike protein. We can see in the uninfected controls there is no detection of that, whereas in the infected cells there is detectable spike protein present near the apical face of the construct, which of course is exactly where Liverpool would have put the virus in the first place. So one of the other things that's important to model is, can we replicate the events that would lead to a cytokine storm, or at least the production of cytokines and chemokines within our model? We use our Mesoscale-cytokine assay, which is employed for a lot of other projects within New Cells Biotech, primarily to do with hepatotoxicity and nephrotoxicity. And if we look at the data from that, we can see that there is secretion of substantial numbers of inflammatory cytokines, things that would be involved in COVID-19 infection and the cytokine storm. Products such as interleukin-6 and interleukin-1b, interleukin-12, and TNF-alpha. Now, in some cases, we see that the secretion of, say, interferon, for example, interferon gamma, is perhaps not as high as would be taking place in vivo. But the fact that we can produce or monitor the production of these cytokines at all, even just at a relatively low multiplicity of infection of 0.01, is encouraging us to think that this could be a useful system to understand the inflammatory implications of viral infection. But also because, you might argue that, okay, we've got a successful method of isolating iPS-derived basal cells. We can make constructs from those which we know are infectable. You could argue that, well, why do we need to do this now? Because there are several companies have asked us if it's possible to use the model to develop new drugs for viruses such as SARS-CoV-2. You might say, well, okay, we've got a vaccine now, or we have 3 potential vaccines coming about. But this is important, what we've done here, because if we can understand the inflammatory consequences of this infection, then we can use our model to potentially model the impact of other viral infections on the lung. And, of course, if we then use our system to look at models such as single-cell sequencing, we can understand the transcriptomic and metabolic consequences of those infections also. And, moreover, we can get back to our original purpose of looking at xenobiotic metabolism of chemicals in these structures. So that's basically the data that I would like to share with you today. It just remains for me to acknowledge some of the staff within my academic group and within various sections of New Cells Biotech, and particularly this individual sitting here, Ivo Jarowski, who is the chap who was principally involved in making these lung constructs. So I'd like to thank you all for listening to this presentation, and I will hopefully answer any questions to the best of my ability that you may have posed. Thank you.